Good. I got a lot of sleep. Pretty good.
I think I'm also missing nice.
Yeah, our own.
I was like, a lot of times I was.
Go ahead. Okay, good. All right, so, hi, everybody. Welcome to the last seminar of the series. Can you hear me? Okay. Hi, everybody. Thanks for coming and to our online audience. So today we are continuing to offer this lecture in a hybrid format with both a virtual and live audience. We are the Department of Biomedical Engineering at Columbia University. I'm Lisa Olson. I'm a professor here. And so our speaker, Adam Engler from UCSD, is going to present for about 45 minutes, and then we'll have some time for questions. And for the virtual audience, please put in your questions at the Q&A at the bottom of your screen, not the chat. And we'll hopefully have time to take some of those questions as well as questions from the audience here. So today we're excited to welcome Professor Engler from the University of California, San Diego. He is a professor and chair of the Department of Bioengineering at UCSD, where he's been on the faculty since 2008. Dr. Engler's current research focuses on how physical and chemical properties of the extracellular matrix influence or misregulate cell function and modify genetic mechanisms of disease. In particular, his lab studies this phenomenon in the context of cardiovascular diseases and cancer. Dr. Engler has received numerous awards in recognition of this research, including Young Investigator or Mid-Career Awards and NIH New Innovator Award, and has been named as a fellow of AIMDE and BMES. So welcome, Adam.
Thank you so much, Lisa, for the kind introduction. All right, so thank you all for attending as well. It's a great privilege to be here. It's been a number of years since I've been back in the city, so it's nice to get away from the surf and all the sun that we've got in San Diego. It was about a 40-degree temperature difference when I got off the plane here, so I can't say that I missed the cold, but one thing that I do want to just tout our own horn is, so I've been chair in San Diego now for about a year, and about a month ago, we were able to announce a naming donation for the department. So I don't usually talk about this, but I just want to put on my chair hat for a second because this just was announced. And so we're very in debt to Jean Lay, who is the CEO of the company called BioLegend. Many of you might get ELISA plates or have antibodies from them. And so his company was sold by to PerkinElmer about a year ago. And so he's made a very generous donation to rename our department the Shu Chen Jean Lay Department of Bioengineering. So I just wanted to toot our own horn about that, but I'll get into my science now and not talk about departmental stuff. I just have a couple of quick disclosures to mention that are science unrelated to what I'll talk about today. I do want to thank those that contributed to the work. In particular, I want to just point out some of the great graduate students, former graduate students now, Matt Ondek, Aditya Kumar, Pranjali Berry did the lion's share of the work that you'll see at the beginning of the talk, and more recent work by former postdoc Katie Birmingham, as well as Madison Kane, Soyoung Moon, and Miranda Barraza contributed to all this. And some of them are highlighted in our photo from our lab get together a year ago, this past summer. So some of those that contributed are circled there. And yes, we've got a lot of palm trees. That's what you're seeing in the background there. So I can talk at length about palm trees. They're terrible. I hate them, but for a variety of other reasons. I do want to also thank the NIH for their gracious funding support from the National Cancer Institute. as well as support from Wyeth Pharmaceuticals. And last but not least, then all of our collaborators, and especially the patients. You'll see some patient data later on in my talk. And so I just want to thank all of our patients. This is where our building is located, the Stanford Consortium for Regenerative Medicine. That is, in fact, the Pacific Ocean. So if you like to surf, San Diego is the place for you to do great science and surfing if you're interested in doing a postdoc or we do have an open faculty search for next year, so definitely be on the lookout for that. Okay, so I think it probably goes without saying to this audience, but I do just want to reiterate that everything I'll tell you about today deals with the extracellular matrix and how cells interact with that. So if you're not familiar with the ECM, it's this three-dimensional fibrillar matrix where cells sit inside of it. So if you think about this building as being an extracellular matrix, we're all the individual cells within it. So the walls, the floors, and the ceilings are things that we can adhere to or stand on, just like cells do the same thing. Okay, and so this environment can portray several really important things. In particular, they can provide topographical cues, so how rough or smooth is the surface? Is it fibrillar or is it a solid material? Is that solid material porous or not, where the cells can sit in it? What is it composed of? Is it mostly fibronectin or collagen? So is this building mostly stone or girders or things like that? And what I'll spend the lion's share of my talk talking about then would be a concept that we have sort of developed over the last 2 decades plus. And that's the difference between something that's really stiff, like the desk, and something that is, unfortunately, after being a faculty member for 15 years, way too soft, like I. So the first sort of foray specifically in cancer and thinking about this was some work that Val Weaver's lab did. First of all, she was at Penn and now she's at UCSF. So a lot of us have this sort of like East Coast to West Coast migration. But one of the things that she pointed out now about 15 years ago was on the left-hand side, you can see that we're changing, or she was changing the amount of collagen that's in the system that these cells are living in. And these are all mammary epithelial cells. And so most of these cells should form nice three-dimensional spherical matrices, as you can see here, right? So they're forming this three-dimensional hollow structure. Now, as the material gets stiffer and stiffer, i.e. their collagen concentration goes up, now all of a sudden, these cells undergo EMT and they form these sheets of cells. So they look much more mesenchymal than epithelial. Unfortunately, though, that's not a clean system because the mechanics and the chemistry are all intercoupled. So we, in fact, use a system that they use called polyacrylamide. It's what you use to separate proteins with SDS page gel electrophoresis. And so again, you can see how you get this very nice sort of three-dimensional hollow spheroid when the material is soft. And then when it becomes stiffer, now you begin to see this transition into a more more two-dimensional sheet. And so this transition is really important, and it happens in a range that is biologically relevant. So if we're thinking about a normal mammary gland, its Young's modulus is somewhere in the neighborhood of about 150 Pascal. However, for tumors, we're now more than an order of magnitude more rigid or stiffer. And so that's why with manual palpation, a woman can actually feel a breast tumor. You can feel, a lesion on your skin if you're developing a melanoma or skin cancer. And so this was sort of the first identification that stiffness really does matter. So we've been thinking about this ever since and have sort of 2 basic observations. I'm not going to have time to tell you about the first one, but a variety of papers are shown up at the top here that I would draw your attention to. But basically, we've been asking, what is the intracellular regulator of stiffness-mediated epithelial to mesenchymal transition? And so the take-home message is that there are a variety of different pathways, and oftentimes those pathways are redundant. And I'll get into a little bit of that because all of these questions, as well as the materials that Val was using, these are all static, meaning that their properties are never going to change once you polymerize that network. However, we wanted to ask the second follow-up question with what happens when cells I should say, form 3D structures before stiffening, or when cells migrate from a stiff tumor to a soft stroma, which is what would normally happen as the cells are disseminating from that tumor. So we're going to use a system, whoops, my apologies, that's a step more advanced from polyacrylamide, and that is this system here. It's a hyaluronic acid with a methacrylated backbone. What this allows us to do is to undergo sequential polymerization. So you can see that we start out at a relatively low Young's modulus, around 100 Pascal, and then we can stiffen that, say, 30-fold or 40-fold up to something that looks like a normal tumor microenvironment might live in, might exist. So we asked two really important questions. The first is, how does acinar development change with different stiffnesses? Again, acinar development means the three-dimensional buds at the end of milk ducts. And then the other question is, how mature do these acini need to be before they become less sensitive to these changes around them? So the first observation then was to put these cells into materials of, a single modulus, so about 150 Pascal, and then stiffen that environment and then discern what the behaviors are. So we're doing sort of a pulse chase assay where the pulse is 150 Pascal for about a week in culture. And then for another 5 days or 10 days, we come back and we stiffen it. So this is just the negative control on the left-hand side, moving over to the positive control on the right-hand side, where we stiffen it to something that's ridiculously stiff for breast cancer. And so if we quantify the cell's behavior, we can see these sort of intermediate stiffness differences. So where you stiffen the environment from, say, 100 Pascal to 1,000 Pascal, now all of a sudden, you're getting about 50% of the cells that don't respond to this cue. So to us, that indicated, and unfortunately it's cut off here with the closed captioning, but the observation that Val's lab had was that this transition actually occurs somewhere around 500 Pascal. So by being dynamic, we're allowing the cells to communicate with each other to form that three-dimensional structure first, and that provides some buffer in terms of their response to the microenvironment. Now, the second question that we wanted to ask was, rather than doing a stiffness gradient, what if we do a temporal gradient? So on the left-hand side, it's just a complicated series of bright field images where we're looking at this 3D hollow spheroid, which in bright field microscopy looks like this phase-dense object. So you can see that very clearly here in like the lower left-hand side of your of the images. And so if we start to stiffen that, hopefully what you notice is in this bottom row, you see most of these cells are staying in these three-dimensional spheroids. And that's because, again, they were mature enough to have enough cell-cell communication to override the stiffness cue. And you can see that in the plot here on the right, where, here we go, where you see a decrease then in the response. So this is the percentage of spheroids that remain, but we have this sort of persistent 20% or so of the cells that are now insensitive to stiffness. So that was really interesting to us. And we wanted to then know, are there any sort of like canonical mechano-sensing pathways? And this does not seem to be, there we go. So are there any of these sort of canonical pathways that we can look at? So we asked a lot of questions related to that. I'm just going to get to the punchline because all of this was published. But if we do the following experiment and we say, let's take these cells, let's put them into a non-dynamic system, we see very nice separation between our stiff microenvironment where the cells are responsive and the soft microenvironment where they're not. So we're looking on the y-axis here at the nuclear to cytoplasmic ratio and on the x-axis too, for two different signaling pathways that are indicative of cancer development. And so the soft microenvironment, everything is relatively cytoplasmic, whereas in the stiff microenvironment, everything is relatively nuclear. Now, if we do the same experiment in the dynamic environment, one thing that I think you'll find interesting is that the differences then become a bit muddled. The acinar cells And the cells that undergo EMT now don't segregate themselves in terms of their nuclear to cytoplasmic ratio. And we think the reason for that is because there are multiple redundant pathways. So if we did these same experiments and we tried to block, say, Twist signaling, or we tried to block YAP signaling or SMAD signaling through TGF-beta, each one of those could be overridden by all of the other pathways. It was only when we blocked all three of these that we could revert the phenotype from what you see on the right-hand side to what you see on the left-hand side. So that was a really interesting observation. But it begged the question then, what happens when these tumors are already established? And we have a very nice collaboration with a group in Brazil. If you've never had the opportunity to go to Brazil, I would highly recommend it. But it is a wonderful environment to do science in because they have some very interesting natural experiments. And one of them happens to be with oral cancer. So the rates of oral cancer are actually 5-fold higher in men than they are in women. And one of the reasons for that is, especially in developing parts of the world, where in some of the interior parts of Brazil, you see these disparities in cancer rates. And that's predominantly because men tend to smoke at higher rates in those parts of the world. They use chewing tobacco at higher rates, but they also have a lot of higher sun exposure by being out and working in agriculture. And so they develop a lot of oral cancers as a result. And oral cancer is a great system in which to study these mechanical changes, in part because if you do a PubMed search for stiffness and oral cancer, oral squamous cell carcinoma, you come up with 8 references, and 3 of them are from our lab. And those 3 were all the first. So anyway, it was it is still a very understudied cancer in terms of its mechanical contributions. So what we first started out in trying to think about oral carcinoma and how these dynamic environments could play a role was we just took lines, oral cancer lines that are more epithelial in nature or more mesenchymal in nature. And we looked at their expression of things like mesenchymal transcription factors. And so in panel A, you see that there's very nice staining differences. You see dramatically different invasion properties in panel B, where the epithelial cells in this air-liquid interface culture never actually penetrate their environment, but the mesenchymal cells are highly invasive. And so we wanted to see, are there differences in, say, stiffness-mediated EMT like we saw with the breast cancer situation? And so if I can direct your attention then to panels C and D, here we're taking these cells, we're putting them into either a soft or a stiff environment, and we're leaving them alone for about a week. And then we say, well, what's their gene expression? How did it change? And as you can see, the gene expression, relatively speaking, for the mesenchymal cells didn't change all that much because they're already mesenchymal. But if we look at these mesenchymal markers in the epithelial cells, now all of a sudden they're going up in a very dramatic fashion. So that began to get us to wonder, are these cells plastic in any way? And if we were to then look at oral carcinomas, these are again from patients from Brazil, where we could take samples of the soft oral tissues that had undergone a transformation. And if we try to stratify patient outcomes in terms of are they low stage, meaning low grade or high grade oral squamous cell carcinoma, and do they have a very organized extracellular matrix? We're using this as an indicator for how stiff the tumors are. So if you look in the organized collagen here, you can see by second harmonics, there's a really, or polarized light microscopy, you can see there's a really robust staining pattern in the tumors that we're calling organized versus those that we're calling disorganized. And so hopefully you can see how patient outcomes then stratify quite remarkably in terms of the patients that have organized versus disorganized tumor collagen. So again, we go back to our system where we're methacrylating the environment, and now we're going to actually degrade it to soften the environment. So that way we can get a curve that goes from something that's relatively soft to something that's relatively stiff, and then back down again. So the experiment's a little complicated, but I just want to walk you through it. So the basic idea is that we want to figure out when the cells transition from a soft environment to a stiff environment, and they begin to imprint what the stiff environment is telling them. So they're undergoing EMT. Then when we take them out of that environment and put them into something that's relatively soft again, like as the cells are disseminating out back into the stroma, do they have any memory of that environment? And so we call this sort of like contractility drives mechanical memory. And by contractility, I'm saying like we can play around with this chemically as well, but here I'm just showing you biomechanics. So the basic idea is that these epithelial cells, when the environment is soft forever, the cells don't move very much, right? And if the environment is stiff forever, like it is in these orange dots, then they don't move, then they move very quickly. They imprint the the mesenchymal pattern, and then they maintain that programming. Now, the cool experiment, though, is what happens when you switch the environments, right? So here, and the control is just our mesenchymal cells, right? So when we go from a soft environment to a stiff environment, so they're imprinting in the soft, so they're staying epithelial, and then we switch them, and we only give them a day, and then we start observing them. They really haven't learned all that much they haven't really undergone EMT yet. But importantly, this category that I'm calling learn, where the environment was stiff, where we stiffened the environment, they sat down, they experienced epithelial to mesenchymal transition, and then we softened the environment. They actually maintain their ability to migrate. So they maintain these mesenchymal markers, which we were really surprised by. So we call this like an environment where they're learning, quote unquote. And we can modulate this also with contractility, right? So we can throw in blebbistatin and screw this up. We can throw in a contractile agonist like LPA, and we can artificially enhance their contractility. And in a soft microenvironment, we can still make them learn if we make them hypercontractile. So then the question was, well, how are these cells actually, what pathways are they using, right? We can look at EMT markers. That's sort of like the end point. That's the output of the black box, and the input is contractility. But what's in that black box, right? So as it turns out, when we did a phosphokinase screen, we said, okay, well, what's coming up differentially speaking? What we ended up seeing, let's get the cursor here. We ended up seeing in the learn environment, the highest expression was for phospho-AKT. And in the recall environment, this is now, once we've softened the environment and these cells need to remember that they're mesenchymal, it turns out that focal adhesion kinase, Y397, was a really important kinase there. So we could repeat all these experiments again, but now instead of just putting them into an environment where they quote unquote learn, or in an environment where we're suppressing their ability to learn with blebbistatin, we can also do that through pathway inhibitors like an AKT inhibitor. And now that don't learn, even in the presence of all of the contractile agonists, doesn't matter. We've shut down the pathway so they don't learn. And we validated that all of this is actually what happens similarly in tumors. So if you look at the tumor core of all of these oral squamous cell carcinoma patients, they have really high expression of phospho-AKT. And the patients, once you get out into the invasive zone around the cells, that phospho-AKT drops. I'm not showing it here, but the focal adhesion kinase goes up. So it seems like there's a switch-like behavior. Now, we were interested in these pathways, but we wanted to take a further step back and just do some RNA sequencing and say, what's globally changing in these cells? Like, what's the transcriptional signature? And so we found about 100 or so genes that help contribute to this difference between the don't learn conditions and the learn conditions. And so we took these transcripts then, and we put them into something called the Cancer Genome Atlas. And if you're not familiar with it, it's this wonderful database that NCI, the National Cancer Institute, has, where you can put in your favorite genes, you can create a Z-score with them, and you can then go into the patient database and say, what were the outcomes of patients that had a Z-score that reflects the don't learn or the learn conditions? And fortunately then, I'm showing you the data because we saw a very good stratification between patients that have these don't learn cues, where they might have like really low AKT, right? So tumors that are low in AKT, they don't learn or they forget. And the patient survival rate's really, really good. But that's not necessarily the case if you have really high expression of AKT and FAK and all of those other transcripts. So that was really kind of eye-opening for us. And what it suggested was that there perhaps is some sort of signature then that we can use to help better diagnose or assess patients. But there's one other part of the story that I haven't really touched on yet, and that is, how does the cell actually remember? So once it's out into the stroma around the tumor, what is it really doing to recall what it has become? So there has to be some sort of imprinting at the epigenetic level. So as it turns out, we can go back to these same patients and we can say, well, what is their expression of an epigenetic marker like a histone 3, H3K9 acetylation, right? So, or H3, oh, that should just say K9 acetylation, right? So if you have really high acetylation, that's actually a signature for cells that are protected. So these patients then have really great survival. And patients that have very low acetylation, meaning that they tend to methylate their histones, have very low survivability with oral squamous cell carcinoma. So this was, again, another sort of intriguing observation. We went back to our RNA sequencing, and we found out that in the LEARN case, again, the patients that don't do very well, they have high expression of a lot of these histone markers as well as methyltransferases, meaning that they like to methylate their histones. And so that's a bad thing. And there's a lot of DNA repair proteins then that get upregulated in the, or downregulated in this LEARN. So we're not repairing our genome properly, and we're methylating it, which is really bad. So we went back to our assays and said, okay, well, what are ways that we can change histone acetylation? So there are some inhibitors out there for methyltransferases, and so when you throw these in, now the key experiment that I want to draw your attention to are these two black curves or sets of data here, where we can downregulate the cell's ability to migrate, so they're less mesenchymal, if you will, in the presence then of inhibitors of these methyltransferases. So hopefully what I've convinced you of thus far is that dynamics are really important, whether we're talking about epithelial carcinomas like breast cancer, where you need to have the right stiffness at the right time for these cells to respond to, or to perhaps not respond to stiffness. And that in the context of let's say a squamous cell carcinoma, that yes, stiffness actually is really important in squamous cell carcinomas, and that it provides some way to imprint on these cells an EMT pathway or an EMT phenotype, which then the cells can remember and recall later once they're invading the microenvironment around themselves. So that being said, I'd like to just take the last, say, 15 to 20 minutes to switch gears and think not about the environment and how the environment is changing cells, but how the cells themselves are responding to that environment. So we're sort of switching from, say, mechanobiology to a bit more biomechanics here. And I think everyone here and online as well can appreciate the concept first of cancer stem cells and the fact that these cells have similar sort of protected behaviors in that they're sort of plastic, they'll migrate out and interact with their environment. Now, unfortunately, there is no really good marker for what defines a cancer stem cell. So this is just a smattering of reviews that I picked out randomly from the last decade, but no one can agree on a single common marker. And many times people can't even agree in one tumor type what is a good marker. So we turned back to physical properties and said, well, maybe there's some sort of physical property of these cells that's really important. And so our assay of choice is going to be adhesion assays, because we think that these can be applied to cells in high throughput and actually give us some really nice data in terms of stratification of these cancer cells. So our very first foray into this about 10 or so years ago was I guess we published a paper like 5 or 6 years ago, was to do something we call a spinning disk assay. So you've got a platform that the cells sit on in the center here, and you apply a radial, or an angular velocity to the cells. So that way it creates a radial shear stress that's dictated by this equation next to panel B here. And so if you know your radial position and you know some properties of the fluid that you're using to spin your cells in, you can then create these very nice plots where unspun cells, the density is relatively uniform. And when you spin them, you get these warmer colors in the center indicating high cell densities and cooler colors at the edges indicating cooler or fewer cells. And so panel D here just very nicely then shows the decay curve that you get from this. So it's cut off by the closed captioning, but low shear stresses are on the left-hand side, high wall shear stresses are on the right-hand side. So very nice, high wall shear stress, all the cells detach. Groovy. All right, so we can take this now, this assay, and apply it to a variety of different mammary epithelial cell lines, some of which I've mentioned before. So the MCF10A cells are relatively, that's what you've seen before, they're relatively benign, they're not malignant. And so they are relatively adhesive, adhesive. So you can see that whether or not PBS and magnesium and calcium, whether or not magnesium and calcium are present in the PBS, you're seeing the same properties for their adhesion. If we switch over to a more malignant but not metastatic cell line like MCF7, there's, again, very little separation between these two cell lines. But if you get to the MDA, MB231s, this is a triple negative breast cancer cell line. Now, all of a sudden, you see there's tremendous cation sensitivity. So if you withdraw these divalent ions now, all of a sudden, the cells detach. Now, all these are different cell lines and different backgrounds. So I show in panel D here the MCF10AT line. This is a RAS transformed cell line that we think is really important as an isogenic comparison between panel A and D. And so you can see, again, pretty remarkable transition in their behavior just by turning on a single oncogene. Now, the reason why these cells are detaching is because in these low cation concentrations, the MCF10A cells here, they are expressing, or they're assembling their focal adhesions in a way that is more or less insensitive or very lightly sensitive to these divalent cations, magnesium and calcium. However, if you look in panel H here, the 231 cells, there's a very dramatic difference between the cells and their ability to assemble focal adhesions in the left-hand side with the divalent cations or the right side without them. Excuse me. Now, the problem with that spinning disk assay is that as the cells detach, they go into this big vat of fluid. It's about 500 mils, and you can't figure out who detached from where. So we turned our attention then to what we call a parallel plate flow chamber. So this is not terribly revolutionary in terms of microfluidics. In fact, our first version was really sort of like a high school science experiment. We held everything together with binder clips. But of course, as engineers, we're much better. We can do it now with microfluidics. We have very large chambers where we can sort about 10 million cells per chamber. We have much smaller chambers now that we can put onto our microscope and we can image them. But these are all straight wall chambers. We can actually make also diverging chambers. So that way, or converging depending on the fluid direction. So this now applies a shear stress that is dependent on the position relative to the cover slip itself. So you can see, we can create COMSOL models and measure the wall shear stresses. But the take-home message is if we apply these shear stresses and we detach cells in different fractions, the most weakly adherent fraction becomes the most migratory. Whether we're looking at breast cancer here in panel A, or on the bottom here, we're looking at a lung cancer cell line, these cells are more migratory, statistically speaking, than their counterparts that are strongly adherent. They also migrate over a longer distance, and so that was great. But taking this system then, looking at the sorted populations and then saying, are these different or how are they different? We again went back to RNA sequencing, and we could see that there are about 500 or so genes that are differentially regulated between these populations. So that's the sort of signature, and we can play around with that in panels D&E and modulate the differences. Now, again, we could move back to the Cancer Genome Atlas. We could take this signature of about 500 or so genes, and we can say, okay, well, is there something unique about the patient outcomes from those that have this signature of being weakly adherent versus those that don't? And again, what was really cool was that we could see differences. So if you're in that strongly adherent group, the blue data here, now all of a sudden you see that most of those patients are going to live, whereas most of the patients that have this weakly adherence signature, their disease-free interval and their progression-free interval are actually really markedly reduced. Now that's just, this is all triple-negative breast cancer. We can also look at invasive ductal carcinoma, and we can look at invasive lobular carcinoma. We get the same sorts of behaviors back out of these systems. So we said, that's all fine and good, but all that data was in vitro and in silico. So what happens when you take these cells and you actually sort them and then put them into mice? So the first sort of foray for us into this was to take these 231 cells, again, a triple negative breast cancer line. We sort them into weakly adherent or strongly adherent. We inject them into a mouse, and then we wait 6 weeks. So the primary tumors that they make in panel C, you can see there's no difference basically in their tumor weight to body weight ratio. Tumor weight by itself doesn't change very much. Tumor volume doesn't change. So they are forming the same sorts of primary tumors. But here's where the differences stop. This is where the similarities stop. So you can see here in the bottom panels, if we were to take out the lungs, and then we count the number of GFP positive nodules. So all these cells have a genetically encoded firefly luciferase expression and GFP expression. And so if we just look at the GFP signal, we see a statistically significant difference between our the number of metastases in the lungs. And if we normalize, because all these mice were sorted in batches, if we take out batch effects, you can see that the sorting is even better. So all of these weakly adherent cells are creating vastly more metastases in the lungs, despite the fact that the primary tumors are relatively the same. Now, we sent this for review. A very astute reviewer said, you're only showing this data in one cell line. What about a different line? Thought that was a great criticism. So we went back and we did that experiment again. And again, we see that weakly adherent cells produce the most tumors in these mice in their lungs, or metastases in their lungs, relative to unsorted or strongly adherent cells. So that's all fine and good. But the model system that we're using, where you just inject the cells, you wait for a certain time, and then you sack the mouse, is not what a patient sees. Right? So a patient would normally come in, they would receive some sort of treatment, they would have the tumor resected, and then there would be follow-ups over a number of years to make sure that the patient was still disease-free. So we said, we need to create a mouse model that mirrors that. So we went back and we said, okay, well, we can take, and I'm sorry that it doesn't reproduce very well, but we can take these cells out at 3 weeks. We can also take a number of mice out of the study and just look at their lungs at 3 weeks. And we can say, okay, well, the lungs were negative when we removed the primary tumors at 3 weeks. So if we were to close the animal back up, right? So let me just walk through the experiment. So we do the injection, right? We remove the primary tumor at 3 weeks. We close up the mouse and we monitor the mouse for another 9 weeks. Now, presumably we got all the tumor, and so we leave the tumor adjacent stroma alone, right? So if there are any dormant cells that are present in that stroma, then they can cause metastases. And so when we do that, this is exactly what we find out. So literally out of 6 mice, there were 2 lung metastases in the strongly adherent group. So we thought these mice, their lungs are totally clear, or almost totally clear. However, in the weakly adherent case, we saw, again, these lungs were lighting up. And this was really surprising to us, but suggested that the weakly adherent cells are capable of remaining dormant in that. First of all, they were capable of disseminating in about 3 weeks from the primary tumor. And then they were capable of laying dormant for a number of weeks before they then metastasized again. So in terms of detection, though, all of this is fine and good, and it tells us that weakly adherent cells are bad, but it doesn't tell us how we can detect it. So we went back to our diverging chamber. So this is the chamber that's not straight walled, but has a diverging pattern to it. So that way, the shear stress we can apply to a single population a variety of shear stresses. And so then we said, okay, well, let's take a monoculture just of the 10A cells or the 231 cells, and let's look at their behavior. And we see dramatically different results between the blue and the green. And then we said, okay, well, what happens if we do an add mixed configuration where we mix the two cells together in a given ratio, and then we fit them not just to a single decay curve, but to a decay curve that has two different sets of constants to it. And here's where it gets interesting. Our predictive ability then, you can see, I think it's very easy to look in the orange data here. So first you have sort of one curve, and then you have sort of a second hump, if you will. And so this sort of bimodal distribution gives us two, or this sort of biphasic distribution gives us two very important sets of properties. The first is it gives us the shear stress where this curve changes shape, but it also then tells us what fraction of the population had detached at the time where this curve changes. So this gives us two really important label-free metrics that we can then use to predict outcomes for patients. Right, so in this case, it's lungs and the number of lung metastases in mice. But so we're normalizing everything here in terms of the predictive capacity of this model. And then what's being plotted here in blue, these are all stromal samples from negative controls. And then what's plotted in red, those are all our experimental group. So did these mice develop tumors or not? And what you're seeing on the x-axis here, that's the wall shear stress. And then on the y-axis, that's the fraction of the population that detached where this y-axis point, x-axis point occurred, where the shear stress at which you saw the concavity change. And so all the red data then occurs in this region where it's highly predictive for metastases. So there's my cursor, right? So this yellow region here, you want to avoid if you're one of those mice, because then you developed a lot of metastases. And so all of these mice then that developed lung tumors were in this high probability range, which tells us then that our assay is pretty predictive. On the right-hand side, this is an area under the curve measurement. So it's a receiver operator curve, if you're ever familiar with those. It's just an indication of how accurate your test was. So it's plotting the false positive rate to the true positive rate. And so a perfect assay would have an area under the curve of 1, meaning that all of the true positives occurred and you detected those, and none of the false positives occurred in your data set. So for us, the area under the curve was 0.94, which we think is a really great result. Okay, so in terms of patients, though, everything that I've shown you thus far has been cell lines, in silico data, or mouse models. So what we were really interested in was getting to our patients. And so here I'm just showing you sort of our pipeline. This is a patient actually from yesterday. So this was a ductal carcinoma in situ patient that came into the breast health clinic at UC San Diego. So we fractionate their tumors into different pieces, different regions that are clinically important as deemed by our surgical colleague. We then fractionate these samples and pull out the cells of interest. So what we get back are, of course, adipocytes, but we also get back cancer-associated fibroblasts and mammary epithelial cells. So we look at those epithelial cells and we put them through this assay. And so here I'm showing you the first dozen or so patients. They come from 3 different arms of our study. So we have triple negative cells. Those are sort of triple negative breast cancer patients. That's sort of like a positive control. And then we have our Reduction mammoplasty, that's a woman that's coming in just for a breast reduction. It could be because of a double mastectomy, but most of the time it's just a straight reduction. And then this is sort of our experimental group, all those patients that have ductal carcinoma in situ, and those are the black data points. I apologize if it's hard to see. I've struggled with how to color the data here. But so these first 10 or 12 patients, one thing that you can see is that all of our negative controls fall within this very low prediction group from our mouse model. And the positive control, the triple negative cells, our triple negative patient is really positive. But I want to point your attention then to all the black data, this sort of experimental group. Now, one of the problems and the reasons why we developed this assay in the first place was because of the problem with the predictive power for ductal carcinoma in situ. So about 1 in every 5 women that develops ductal carcinoma in situ will not remain cancer-free. Her cancer will reoccur somewhere within the 3 to 5 year window, which is really unfortunate. And it's a problem of treatment. These patients, because of the histological markers that are currently used to diagnose ductal carcinoma in situ, these patients were missed. They had some form of the disease of DCIS that was much more aggressive, and the treatment plan that was prescribed by the provider was insufficient in maintaining the health of that woman. So we're numbers-wise, we're talking about 10,000 or so patients every year who have their tumors reoccurred, even though they had a very regionally invasive, very focal disease that otherwise a stronger treatment plan would have prevented from reoccurring. So these are like think of this as 10,000 patients that unnecessarily are getting secondary disease. And so we think that this is an assay that, if applied to all women that had ductal carcinoma in situ, we could catch this 20%. So 10,000 patients might not seem like a lot, but in the grand scheme of things, if we can prevent one of these patients from reoccurring, we think we've done our job. So in the interest of time, I'm just going to highlight 2 very quick last slides before I go to my acknowledgements. But I've had a great collaboration over the last couple of years, moving away from sort of the diagnostic angle for this assay, but really trying to understand from a mechanical perspective, how can these cells migrate against a stiffness gradient? So if you're familiar with the concept of durotaxis, that's the idea where cells move from something that's soft to cells move to something that's stiff. And so 90% of our adherent cells in the body will do that. You know, muscle, bone, you name it. They durotax. But there's one exception, if you think about it, and that would be cancer. These cells have to go from a stiff tumor to a softer microenvironment in the stroma that surround them, just like we saw with the oral squamous cells carcinoma. So we developed this, we call it a zebraxis pattern, where it's stripes of soft and stiff matrix. My postdoc a decade ago thought this looked like stripes on a zebra, but biomaterials didn't like us coining that phrase. This was back before Cam was the editor. But I want to close with this slide because I think it's really a poignant depiction of this idea, where if you take these cells, normally at 0 hours, they have a very even distribution across the surface of this substrate. But if you let them sit for, say, 24 hours, cells begin to accumulate in these stiffer stripes if they are strongly adherent. Those are the cells, again, that were not very aggressive, right? They didn't move very much. We generally think that they stay in the tumor. The weakly adherent cells, the ones that are disseminating, they don't undergo durotaxis. In fact, we have evidence that they undergo more anti-durotactic or adurotactic behavior. So the weakly adherent cells are greater there. I lied, I've got one more slide. Here, we did the same experiments, not just with an epithelial cell from breast cancer, but we also looked at lung carcinomas on the right-hand side and prostate cancer on the left-hand side. And again, we see a higher portion of these cells are being anti-durotactic or they're adurotactic, meaning that they migrate down the gradients or they oscillate back and forth, where sort of the net migration of these cells tends to be that they move away from the tumor. So with that, I just want to thank everyone for their attention today, and just conclude by saying, if you took home nothing from the second half of what I told you about in terms of adhesion strength, hopefully you'll take home that this is a really important, we think it's a really important biophysical marker that can be used to separate cells and that potentially could be used as a diagnostic tool in the future to help better detect these sorts of really subtle cancers that are really hard to determine treatment plans for, like a ductal carcinoma in situ. So with that, I just again want to thank everyone and say, I am beholden to the quality of grad students and postdocs in my group. So they love doing fun things like, I don't know, they said this guy, Chris, just showed up everywhere. So as these cells are dividing, Chris's head pops up because he's like metastasizing everywhere. Anyway, I digress. Thank you so much for your attention. It's been a lot of fun and thanks for being here.
So I'll just say again, thank you to Adam Engler for such an interesting talk and very important material. So I am now just going to alert you that you can follow us on social media to receive the latest updates. The slide is up there. And we will be back in January 27th. So happy holidays to everybody. And now we have time for questions. And do we have any questions from? Okay, great. And can you repeat the question?
Sure. Yeah, no problem.
So I saw that in the hydrogel model, you use here as a photoinitiator. Have you explored any other photoinitiators? Because I've heard that there's some cytotoxicity from.
Very astute observation. Yes. So we tend to use Ergocure a lot. We also use LAP. I don't think I've got the slide with me, but that was a really important consideration. We had this PNAS paper in 2019, and one of the reviewers said, well, are all these observations just due to the fact that there's some UV present in this Ergocure? So we did a combinatorial experiment where we exposed the cells to just Ergocure or just UV, and then both. We didn't see any differences in the cell behavior long-term out to like 2 weeks. We're only using about 90 seconds worth of UV. However, if we do sort of a time course and we say, okay, well, now let's expose them from 30 seconds, 60, 90, 5 minutes, a half an hour, we see DNA damage occurring at about the 30-minute time point, specifically with the MCF10A cell line. So we haven't gone back and verified that with every other cell line that we've used the system in, which we probably should. But yeah, so the question was, about the photo initiator that we're using, which is Ergocure, and so yeah, thank you very much. That's a really important point.
So, do we have any other questions from students? Because we have a few online questions, a lot of them, in fact. But I don't want to run over. Now the clock is gone. What time is it? Okay, great. So we have time for some questions. So I guess I'll just go in order, or if you want, you could come read along, but I'll, so Ahmet Topal asks, how did you look at and measure the cell stiffness and determine the stiffening throughout the post-morphogenesis days?
Right. So the question about stiffness is that we're actually changing not the cell's stiffness, but we're modifying the matrix that they're attached to. So we measure that by atomic force microscopy. So I've got an AFM in our lab, and we just measure that in the cultures. Now that's a surface technique. So we're measuring the top surface of these three-dimensional cultures. That's not to say that there could be some differences as a function of Z, but we have not seen differences in cell behavior as a function of Z in these cultures. So if there is some differences, the cells at least are not responding to it, and they're being relatively uniform in their response, depending on whatever the modulus is.
Okay, I'll ask again if anybody has questions here, or otherwise there are 13 questions here. We won't do all of them, but okay. So I'll go on to Grace Hinz is asking, how do non-malignant tumors that also exhibit an increased stiffness play into your understanding of the relationship between stiffness and cancer? So non-malignant tumors.
Very good. Yeah, very good question about malignancy. And so there are, I think there are sort of like two two things that I don't think that we really appreciate. So what happens in a malignant but non-metastatic tumor? And then the flip side is what happens in breast tissue that happens to be very mammographically dense. So it's very radio dense. Those tissues can also be relatively stiff, but are not tumorigenic. So we're trying to tease out like what are the specific pathways that are being turned on in those particular instances. And so part of our tumor surveillance program is to try to get the reduction mammoplasties from women that have, say, high mammographic density and understand those cells and their behavior. That's actually what our funding is from Wyeth, is to look at mammographic density and what happens in these sort of outlier cases that don't follow these rules. So unfortunately, the answer is to be determined.
So I'll ask another one from Grace Hines, because I thought this was an interesting question. Do you see any therapeutic potential to target weakly adherent cells?
Yes. So we currently have a drug candidate from the second of my disclosures, Xenon Pharmaceuticals. They are interested in, they have a small molecule inhibitor that is directed at focal adhesion kinase, and we think that that might actually play a role in mechano-sensing. So currently all that data is proprietary for the company, but I think it's very promising that we could target the signatures of these weakly adherent cells and use the differences in those signatures then for potential drug targets. So we think that we've got one with the Xenon company.
Okay, here's another sort of related question, which may be, also something you can't tell us, but Yang P. Louis is asking, does chemotherapy change the adhesive property of the dormant stroma cells?
Ooh, definitely can answer that. The answer is yes, absolutely.
Oh.
So that's, we have been working with Cindy Reinhardt King. She several years ago showed that matrix itself can be, its mechanical properties can change with exposure to radiotherapy. It's a structural rearrangement of the collagen due to the damage that it gets through the radiation therapy. So we've been going back and looking at irradiated collagens and how cells that are placed on these irradiated collagens, weakly adherent versus strongly adherent, can change their behaviors. The take-home message is, one, again, that data is still pretty nascent, but we think that there is a difference and that it prevents the cells from migrating as fast. So weakly adherent cells can stay weakly adherent, but they can't migrate very well.
So we're sort of out of time, but I'm going to ask one question of my own. So is the fact that as people age makes their tissues get stiff, is that part of the reason that maybe we develop cancer when we're older, or?
I think it's so multifactorial that it's hard to tease out any one particular cause, other than, say, some of the obvious ones, exposure to carcinogens and things like that. Genetic makeup, again, being really important. But we certainly do see, older populations that are developing this. And if you look at just tissue properties over time, that those will increase. So just sort of natural physiological aging, we do notice a difference. And so I would say that while I don't know if there's any definitive data, I would be surprised if that wasn't a contributor.
Okay, so let's thank our people.